Our next topic is defecation. So we'll start by talking about the mass movements that force feces toward the rectum. And these mass movements distend or stretch the rectum and initiate the spinal defecation reflex. These involve parasympathetic signals that stimulate contraction of the sigmoid colon and rectum and then relax the internal anal sphincter. We are allowed conscious control, however, over the relaxation of the external anal sphincter. The muscles of the rectum contract to expel feces, and they're assisted by Valsalva's maneuver. This includes closing the glottis, contracting the diaphragm and the abdominal wall muscles, but this increase in intra-abdominal pressure is able to help expel feces or force defecation. The levator anal muscle also is going to contract, and the anal canal will be lifted superiorly and allow feces to leave the body. So let's look at the defecation reflex in more detail. Here we can see the sigmoid colon and the rectum, and as feces moves into the rectum from the sigmoid colon, it would stretch or stimulate stretch receptors. And then we would see that those receptors are able to transmit signals considered afferent or sensory signals to the spinal cord. Those afferent or sensory signals to the spinal cord are being carried by parasympathetic nervous system fibers, and then we will get a motor response or efferent response back from the spinal cord, and this spinal reflex would cause parasympathetic motor efferent fibers to stimulate contraction of the rectum and the sigmoid colon, and eventually relaxation of the internal anal sphincter. So it really is a reflex. We're seeing the parasympathetic nervous system primarily being used, and we're seeing that the receptor is a stretch receptor. The stimulus of stretch is sent along an afferent neuron to the central nervous system. The central nervous system is going to use the spinal cord as its integrator, and we will send the signal from an input neuron to an output neuron, which is motor or efferent, and that signal will then go to the internal anal sphincter and cause its relaxation. And then last but not least, if it's convenient to defecate, we will voluntarily relax or inhibit motor neurons to the external anal sphincter, and this will allow feces to pass out of the body. So that's the defecation reflex. Because of the external anal sphincter being skeletal muscle and the internal anal sphincter being smooth muscle, we have both a reflex and voluntary inhibition of the external anal sphincter. Chemical digestion in the entire GI tract involves the use of catabolic reactions that break macromolecules down into monomers. Monomers, or small subunits, are small enough for absorption. We're also going to use enzymes, and we're going to use hydrolysis, or water, to break bonds. We'll break down carbohydrates to monosaccharides, and enzymes we'll use to do so include salivary amylase, pancreatic amylase, and brush border enzymes. They'll break down polysaccharides to oligosaccharides, and then oligosaccharides to disaccharides, and then finally disaccharides to the monomer monosaccharides. Starch is an example of a polysaccharide, and its digestion would begin in the mouth. All carbohydrate digestion begins in the mouth with salivary amylase. Salivary amylase can break down polysaccharides to oligosaccharides, but those oligosaccharides must travel to the small intestine and be acted upon by pancreatic amylases in order to be broken down into even smaller oligosaccharides or disaccharides. Then brush border enzymes are able to break down disaccharides or smaller oligosaccharides to monosaccharides. I highly recommend studying the flowcharts of digestion over the different foodstuffs. Here we can see that we're looking at the digestion of carbohydrates. So we begin with, say, starch or disaccharides, and we're going to end with monosaccharides. Carbohydrate digestion begins in the mouth and ends in the small intestine and uses salivary amylase, pancreatic amylase, and brush border enzymes in order to digest carbohydrates to their monomers. Next, we're going to look at the digestion of proteins, and the digestion of proteins begins in the stomach thanks to pepsin. Pepsin is activated from pepsinogen because of the hydrochloric acid that's located in the lumen of the stomach. So pepsin is actually inactivated as it moves from the stomach to the small intestine because there's a much higher pH in the duodenum. We're going to use pancreatic proteases and brush border proteases in order to break down proteins. We'll see that throughout the digestion of the different nutrients, they have to be small enough to be absorbed in the enteric epithelial cells. They're also going to be absorbed through the enteric epithelial cells by using ion channels. Some of these ion channels are active, others are passive, so we'll see a mix of primary and secondary active transport and diffusion for the absorptive properties of these nutrients, not only through the enteric epithelial cells, but then into the blood or lymph. The digestion of proteins begins in the stomach with pepsin and ends in the small intestine with our brush border enzymes. We're going to break down proteins to their amino acids, and amino acids are the monomer of proteins. For the digestion of lipids, we need a pretreatment step. So lipids are a little harder to digest. So lipid droplets like to combine together and form really big fat globules. So we have to pretreat or emulsify fat by using bile salts. So it's not going to break bonds, but it's going to make it easier for pancreatic lipases to act on lipids. So what we're seeing here in this slide is the emulsification step. So here we have a big fat globule, and we're going to use bile salts from bile that came from the gallbladder to break down our big fat globule into smaller fat globules. It's a lot easier to act with enzymes on these smaller fat globules than it is to act on the big fat globule. By breaking down the fat globule, we've increased the surface area that the enzyme pancreatic lipase has to act. Once we break down these small fat globules into monoglycerides and fatty acids, we'll use the remaining bile salts to form a structure called a micelle. Micelles will be absorbed through the enteric epithelial cells, turned into then chylomicrons, which will then go into a lacteal or a specialized lymphatic capillary. So just remember that lipids must be pretreated. Emulsification is the process of pretreatment, and it makes it easier for lipids to be digested by enzymes. The emulsification step doesn't begin until we hit the small intestine, so even lingual lipase from salivary glands or gastric lipase from the stomach isn't really doing much digestion until they go into the small intestine, and then along with pancreatic lipases, they're going to help break down lipids. But we have to wait till that emulsification step that occurs in the small intestine. But the first lipase we get added to the GI tract is from the mouth. The last ones will be from the small intestine, and we end lipid digestion in the small intestine after emulsification. For the digestion of nucleic acids is a little simpler. There's not a whole lot. It begins and ends in the small intestine, and we'll break down nucleic acids into nucleotides using nucleases. There's deoxyribonucleases and ribonucleases. They break down different types of nucleic acids, and some of them come from the pancreas, and some are in that brush border or the microvilli of the enteric epithelial cells. What makes up a nucleotide, which is the monomer of a nucleic acid, are nitrogenous free bases, pentose sugars, and phosphate ions. So nucleases are also able to break down nucleotides into these three components. So a pretty easy chart or flow chart for the digestion of nucleic acids, and that is that it begins in the small intestine and ends in the small intestine. And we're going to break down nucleic acids into the components of nucleotides: the sugars, the nitrogen-containing bases, and the phosphate ions. Next, we're going to look at the absorptive properties of the small intestine. So all food and 80% of electrolytes, most water, all of that's absorbed in the small intestine, and most prior to the ileum. So we're going to see most absorption occur before food or chyme reaches the ileum. But the ileum does reclaim bile salts, so it is involved in the enterohepatic recycling of bile salts back to the liver and gallbladder. Most things are going to be absorbed through the enteric epithelial cells by active transport, and they're going to be absorbed via active transport into the blood, all except for lipids. So lipids are the exception. They are not absorbed through the enteric epithelial cells into the blood. Instead, they're going to be absorbed into lacteals. Lacteals are specialized capillaries, and they are part of the lymphatic system. We're going to look briefly at some extra information about the absorption of carbohydrates and proteins and whatnot. Here we can see that carbohydrate absorption uses both secondary active transport and facilitated diffusion. Protein absorption uses several transporters, including those that are going to be able to be co-transporters of other molecules and are located in the apical and basal layer of the enteric epithelial cells. A homeostatic imbalance we want to mention briefly is that the mucosa of newborns is not fully mature, and it allows them to take in whole proteins. Now, we're not normally allowed to take in whole proteins into our mucosa. They're broken down to their amino acids, and that's what is absorbed. So the taking in of whole proteins by the GI tract mucosa actually can cause food allergies in newborns, which disappear as their mucosa matures. But luckily, there's passive immunity offered to them by the breast milk of their mothers, which produces or has in it immunoglobulin A or antibodies. So just a homeostatic imbalance that relatively rare but is seen. For the absorption of nucleic acids, just know that it requires active transport across the epithelium, and we know that our carbohydrates, our nucleic acids, our proteins, they are being absorbed into the blood, whereas absorption of lipids is into lacteals, when we also see in the small intestine that anything that is lipid soluble, like our fat soluble vitamins, doesn't even need a transporter. It's able to go straight through the plasma membrane. So vitamins such as A, D, E, and K are just carried by micelles and diffuse into the absorptive cells without any channels or carriers. Whereas our water soluble vitamins have to be absorbed by diffusion or by passive or active transporters. Vitamin B12 is actually too big to be absorbed with passive or active transporters, and it can't go just straight through the plasma membrane. Instead, it has to be absorbed by endocytosis. And don't forget that there is some vitamin K and B made by the bacterial metabolism in the large intestine, so it's not all absorbed in the small intestine. And some electrolytes are absorbed. Most are absorbed in the small intestine. Only the remaining ions that make it to the large intestine may be absorbed. Commonly, that would be sodium and chloride. And then iron and calcium absorption is going to be related to need. So if we don't need it, it remains in the large or small intestine and is ferried out of the body. Calcium absorption is going to be regulated by vitamin D and parathyroid hormone. Calcium absorption is one way to allow for the processing of vitamin D into calcitriol or its active form. Next, we'll look at the absorption of water. Most water is going to be absorbed in the small intestine via osmosis. Remaining water would be absorbed in the large intestine. And then our next topic is the malabsorption of nutrients. And anything that interferes with the delivery of pancreatic juice or bile or damages the intestinal mucosa can cause malabsorption. Damaging the intestinal mucosa causes malabsorption because this decreases the surface area for absorption in the small intestine. This could occur because of antibiotics or bacterial infection, or because of a homeostatic imbalance such as celiac disease. Celiac disease is also known as gluten-sensitive enteropathy. It is an immune reaction to gluten, and this gluten immune reaction damages the intestinal villi and microvilli and actually causes a decrease in the surface area for absorption in the small intestine. But we can treat this by eliminating gluten from the diet of an individual who does suffer from gluten-sensitive enteropathy, and this includes all grains except rice and corn. And that is it for part C of our digestive system. So make sure you review all three parts, A, B, and C, now posted.